Welcome to Noun Tutorial TV. Like, share and subscribe. NSC223, Medical Biochemistry 1 Exam Summary. Dash is the study of chemical processes in living organisms. Biochemistry. Dash can be defined as the application of chemistry to the study of biological processes in living organisms. Biochemistry. Biochemistry is both a life science and a chemical science. True. Dash field of science explores the chemistry of living organisms and the molecular basis for the changes occurring in living cells. Biochemistry. Medical biochemistry can be described as a branch of dash. General biochemistry. Dash is defined as the study of biochemical processes that occur within the human body in relation with their application in the field of medicine. Medical biochemistry. A scientific expert in the field of biochemistry is called a biochemist. The study of chemical processes in living organisms is called biochemistry. Dash can be defined as the application of chemistry to the study of biological processes in living organisms. Biochemistry. Biochemistry is both a life science and a chemical science. True. Biochemistry explores the chemistry of living organisms and the dash basis for the changes occurring in living cells. Molecular. Dash is defined as the study of biochemical processes that occur within the human body in relation with their application in the field of medicine. Medical biochemistry. DNA stands for deoxyribonucleic acid. Deoxyribonucleic acid, DNA, was discovered by James Watson and Francis Crick in 1953. Biochemistry deals with the structures and functions of cellular components such as proteins, carbohydrates, lipids and nucleic acids, collectively known as biomolecules. Cellular components such as proteins, carbohydrates, lipids and nucleic acids, collectively known as biomolecules. The main focus of biochemistry is understanding how biological molecules give rise to the processes that occur within living cells. Medical. One of the major breakthroughs in medical biochemistry was the discovery of an accurate model of deoxyribonucleic acid, DNA, by James Watson and Francis Crick in 1953. The following are the branches of biochemistry: toxicology, enzymology, molecular biology and biotechnology, lipid and carbohydrate biochemistry, natural products biochemistry. Discovered deoxyribonucleic acid, DNA. James Watson and Francis Crick. James Watson and Francis Crick discovered deoxyribonucleic acid, DNA, in 1953. The human genome was mapped completely in the year as a result of the 13-year Human Genome Project, HGP. 2003 studies the adverse effects of toxic or foreign chemical substances on the organisms. Toxicology. Is the study of enzymes, their functions, deficiency, and the consequence of such deficiency in diseases. Enzymology. Involves manipulation of DNA to improve drug research and solve health problems. Molecular biology and biotechnology. There are main branches of biochemistry. 5. Medical biochemistry provides foundation for other life sciences such as medicine, nursing, pharmacy, zoology, microbiology. The branches of biochemistry include these: toxicology, enzymology, molecular biology and biotechnology, lipid and carbohydrate biochemistry, natural products biochemistry. Is the study of the adverse effects of toxic or foreign chemical substances on the organisms, environmental and food toxicology. Toxicology. The study of enzymes, their functions, deficiency, and the consequence of such deficiency in diseases is known as enzymology. Lipid and carbohydrate biochemistry study the biochemical basis of metabolic disorders such as diabetes, obesity, and cardiovascular diseases. Dash are monomeric unit through which the complex human body was constructed. Cells. The human body contains several dash cells. Billion. A dash is defined as the fundamental unit of life, and it is the smallest unit capable of exhibiting the characteristics of life. Cell. Cell was accidentally discovered in dash dash. 1665 cell was accidentally discovered in 1665 by dash dash. Robert Hooke. Dash observed numerous porous structures, dead cells made of cellulose found in plants, and named them cells. Latin, cellulo means little room or small chamber. Robert Hooke. Dash are the monomeric unit through which the complex human body was constructed. Cells. In cells, energy generation takes place inside the dash dash. Mitochondria. A dash is defined as the fundamental unit of life, and it is the smallest unit capable of exhibiting the characteristics of life. Living cell. Cell was discovered in dash dash. 1665 cell was discovered in 1665 by dash dash. Robert Hooke. Who observed numerous porous structures, dead cells made of cellulose found in plants, and named them cells. Latin, cellulo means little room or small chamber. Robert Hooke. In Latin, the word cellulo means dash dash. Little room or small chamber. All living things are composed of cells. True. Fig 2.1, Structure of animal cell. Red blood cells are also called dash dash. Erythrocytes. Red blood cells are also called dash dash, erythrocytes. Dash are one of the smallest animal cells, while ova are among the largest. Red blood cells. The thin cover that separates a cell from its environment, it also protect the components of the cell from leaking, is called dash dash. Plasma membrane. Dash prevents the fluid outside the cell called extracellular fluid, ECF, from mixing with the fluid inside the cell called intracellular fluid, ICF. Plasma membrane. Dash regulate the materials that enters or leaves the cell. Plasma membranes. Animal cells have different shapes and sizes, some are circular, spherical, cylindrical, and fibrous. ECF means dash dash. Extracellular fluid. Plasma membranes is semi-permeable. True. Dash is the fluid-like space between the plasma and nuclear membrane, and where the cavity where the organelles are found. Cytoplasm. Dash is the cavity where the organelles are found. Cytoplasm. Dash provides space for the movement of synthesized products from one compartment to another for further processing. Cytoplasm. The organelles are suspended in the dash by cytoskeleton network that resemble nets. Cytoplasm. The organelles are suspended in the cytoplasm by dash that resemble nets. Cytoskeleton network. The organelles are suspended in the cytoplasm by cytoskeleton network that resemble dash dash. Nets. Animals, plants, and fungi belong to the group dash dash. Eukaryotes. Eukaryotic cells are much larger than prokaryotes. True. There are about 210 distinct human cell types, and there are between 50 and 100 trillion cells in adult human body. Animals grow as a result of dash and cell enlargement. Cell division. Animals grow as a result of cell division and dash dash. Cell enlargement. Animals grow as a result of dash and dash dash. Cell division and cell enlargement. All animals begin their existence as a simple cell called dash dash. Fertilized egg. The largest cell in man is dash dash. Ovum. The smallest cell is dash dash. Red blood. There are dash types of tissues in human. 4. The following are the types of tissue. Epithelium tissues. Nervous tissues. Muscle tissues. Connective tissues. Multicellular organisms consist of several dash dash. Organ systems. Organisms consist of several organ systems multicellular. Taste receptor cells in the mouth live for days. 10 normal red blood cell lives for about months. 3 to 4 sickle-shaped red blood cells live for days. 10 to 20 is the most important part of the cell, centrally located. Nucleus. Contains the genetic materials, DNA and RNA. Nucleus. Regulates the rate and time of cell division and determines the materials that enter or exit the cell. Nucleus. The following are the functions/features of nucleus in a cell. It is the most important part of the cell. The nucleus is always centrally located. It has its own membrane called nuclear membranes, which protects the content of the nucleus. Nucleus contains the genetic materials, DNA and RNA. It determines the materials that enter or exit the cell. Differences between prokaryotes and eukaryotes cells. Eukaryotes have nucleus which is covered by nuclear membrane. Animals, plants, and fungi belong to the eukaryotes. Prokaryotes have no typical nucleus, no true nucleus. Bacteria and blue-green algae belong to the prokaryotes. Eukaryotic cells are much larger than prokaryotes. They also have a variety of other membrane-bound organelles in their cytoplasm. Animals grow as a result of and cell enlargement. Cell division. Animals grow as a result of cell division and Cell enlargement. Animals grow as a result of Cell division and cell enlargement. Is the largest cell in man. Ovum. The smallest cell found in man is called red blood cell. Organisms which are able to use specialized cells to perform specific functions are called multicellular organisms. A group of cells is called a tissue. The four basic types are the following: epithelium tissues, nervous tissues, muscle tissues, connective tissues. Multicellular organisms consist of several systems. Organ. Cells within the human body have different lifespan based on the type and function of that cell. True. Taste receptor cells in the mouth live for 10 days, 1 month for the skin cells, 15 years for muscle cells, and a lifetime for nerve cells. The plasma membrane mainly consists of lipids and proteins. The plasma membrane mainly consists of lipids and proteins. The plasma membrane mainly consists of and proteins. Lipids. The fundamental building blocks of cell membranes are the phospholipids. Membrane lipids are molecules. Amphipathic. Hydrophilic means water-loving. Hydrophobic ends means water-hating. Fig 2.2, simplified structure of plasma membrane bilayer. Are the most abundant membrane lipids. Phospholipids. Phospholipids are the most abundant membrane lipids which have a polar head group and hydrophilic hydrocarbon tails. 2. The most abundant membrane lipids are phospholipids. The fundamental building blocks of cell membranes are the phospholipids. Membrane lipids are amphipathic molecules. The major phospholipids predominate in the plasma membrane of many mammalian cells are phosphatidylcholine, phosphatidylethanolamine, phosphatidylserine. Phosphatidylserine carries a charge, negative. Phospholipids such as the inositol phospholipids, which are present in smaller quantities, are referred to as phosphoglycerides. And glycoprotein are found on the outer monolayer of the lipid bilayer. Glycolipids. Glycolipids and are found on the outer monolayer of the lipid bilayer. Glycoprotein. When animal cells undergo phosphatidylserine, which is normally confined to the cytosolic monolayer of the plasma membrane, rapidly translocates to the extracellular monolayer. Apoptosis. Provides stability to the membrane. Cholesterol. In the erythrocyte membrane, The outer membrane leaflet is a rigid ringed molecule with a tiny hydrophilic end, hydroxyl group. 4. In the membrane, the outer membrane leaflet is a rigid four-ringed molecule with a tiny hydrophilic end, hydroxyl group. Erythrocyte. Glycolipids are containing lipids. Sugar. Are sugar-containing lipids. Glycolipids. Glycolipids are sugar-containing lipids. The simplest glycolipid is called cerebroside. The can be used to deliver drugs to target cells or DNA to specific cells for gene therapy. Liposome. Research is ongoing on the use of liposome for cancer chemotherapy. The major phospholipids predominate in the plasma membrane of many mammalian cells are phosphatidylcholine, phosphatidylethanolamine, phosphatidylserine. Phosphatidylserine carries a charge, negative. Phosphatidylcholine and phosphatidylethanolamine are electrically neutral at physiological pH. Phospholipids such as inositol phospholipids present in smaller quantities are referred to as phosphoglycerides. Is another group of phospholipids which contains a sphingosine as backbone. Sphingomyelin. Sphingomyelin is another group of phospholipids which contains a as backbone. Sphingosine. Sphingomyelin is electrically at physiological pH. Neutral. These are membrane lipid found in the plasma membrane of mammalian cells. Lipid bilayer. Cholesterol. Glycolipids. Lipid vesicle or liposome. Membrane proteins. Glycoproteins. The exposed on the cell surface serves as a signal to induce the macrophages to ingest and digest the dead cell. Phosphatidylserine. Provides stability to the membrane, and it is neutral at physiological pH. Cholesterol. The characteristic of cholesterol are the following. It exists in the plasma membrane of mammalian cells. It provides stability to the membrane. It is neutral at physiological pH. The outer membrane leaflet is a rigid four-ringed molecule with a tiny hydrophilic end, hydroxyl group. The simplest glycolipid is called cerebroside. The ability of phospholipids to form lipid bilayer has been used to create an important clinical tool called liposome or lipid vesicle. An aqueous compartment enclosed by a lipid bilayer is known as liposome. Perform most of the specific functions of the membranes. Membrane proteins. Give each type of membrane in the cell its characteristic functional properties. Membrane proteins. In the 75% of the membrane component is protein due to the presence of many enzymes and electron pumps. Mitochondria. In the mitochondria, percent of the membrane component is protein due to the presence of many enzymes and electron pumps. 75 are attached to glycoprotein and glycolipids on the surface of red blood cells. Carbohydrates. Carbohydrates are attached to glycoprotein and glycolipids on the of red blood cells. Surface. Carbohydrates are attached to glycoprotein and glycolipids on the surface of red blood cells. Fig 2. 5. The structure of a cross-section of liposome. Is an aqueous compartment enclosed by a lipid bilayer. Liposome. Membrane proteins perform most of the specific functions of the membranes and give each type of membrane in the cell its characteristic functional properties. Carbohydrate are covalently attached to proteins to form glycoprotein. The carbohydrates of glycoprotein determine the blood group antigens that have been used in blood typing. Carbohydrates are attached to and glycolipids on the surface of red blood cells. Glycoprotein. The blood group common in oligosaccharide foundation is called the O antigen. People in blood group A have one extra monosaccharide called N-acetylgalactosamine. People in blood group B contain an extra monosaccharide called galactose. The dash and the organelles carry out all the functions performed by living cells. Cell membrane. The cell membrane and the dash carry out all the functions performed by living cells. Organelles. The dash can be referred to as the wall of a city. It protects the components of the cell and also regulates what enters or leaves the cell. Cell wall or plasma membrane. The types and functions of organelles. Rough endoplasmic reticulum, synthesis of protein, due to the presence of ribosomes, and degradation of worn-out organelles. Smooth endoplasmic reticulum, synthesis of lipids and steroids, storage and metabolism of calcium and detoxification of toxic substances. Golgi apparatus, processing, packaging, labeling, and delivery of proteins and lipids to their destination within cell. Lysosome, degradation of macromolecules, worn-out organelles and the removal of excess secretory products. Peroxisomes, detoxification of hydrogen peroxide and other radicals because it contains antioxidant enzymes such as peroxidases and catalases. The organelle in the cell which synthesis proteins are called ribosomes. Controls all the cellular activities, absent in matured red blood cells, DNA and RNA synthesis and storage of genetic information. Nucleus. The functions of plasma membranes. Protection. Transportation of materials across the cell membrane. Passive transport facilitated diffusion. Active transport. Is responsible for the maintenance of shape and size of cells. Plasma membrane. Passive transport in cells involves the process of diffusion. Diffusion can be or facilitated. Simple. Diffusion can be simple or facilitated. Diffusion can be simple or facilitated. Is a transport process that requires energy to transport materials across the membranes. Active transport. There are types of active transport. 2. An example of primary active transport is sodium-potassium pump, Na plus K plus At posse. The energy required for the transportation of sodium and potassium ions are derived from the of ATP. Hydrolysis. When materials can be exported from the cell by fusing vesicles with the plasma membrane, it is referred to as Exocytosis. The different types of endocytosis processes that occur in the cell include pinocytosis, receptor-mediated endocytosis, phagocytosis. The cell or plasma membrane can be referred to as wall of a city. Co-transport may be or symport or antiport. A moves two molecules in the same direction, e.g. sodium-glucose transporter. Symport. A moves two molecules in the same direction. Symport. Antiport system moves two molecules in direction. Opposite. Active transport requires carrier protein active sites. 2. Active transport is classified into two types according to the source of energy used. Derives its energy directly from the hydrolysis of ATP. Primary active transport. Primary active transport derives its energy directly from the hydrolysis of ATP. The uses an indirect energy of an electrochemical gradient or membrane potential produced originally by primary active transport. ATP. System moves two molecules in opposite direction. Antiport system. Biochemistry is concerned with the chemical components of the human body, their properties, functions, synthesis, transport, and degradation. The protein or enzyme responsible for the transportation of Na plus and K plus across the cell membrane is known as sodium-potassium adenosine triphosphatase. The energy required for the transportation of sodium and potassium ions are derived from the hydrolysis of ATP. Movement of materials of the cell involving changes of the membrane and formation of vesicles is called exocytosis. The movement of materials into the cell is called endocytosis. The major classes of biomolecules include these: carbohydrates, lipids, proteins, nucleic acids. The building blocks of biomolecules are the following: simple sugars, fatty acids, amino acids, mononucleotides. Are compounds containing C, H, and O and having the general formula CnH2 non. Carbohydrates. Is the most important carbohydrate. Glucose. The major metabolic fuel of mammals and a universal fuel of fetus is glucose. NB. Carbohydrates are widely distributed in plants and animals where they play important structural and metabolic roles. The diseases associated with carbohydrates metabolism include diabetes mellitus, galactosemia, glycogen storage diseases, lactose intolerance. Fig 1.1, Biomolecules. Carbohydrates are classified according to the number of sugar units in the molecule as follows: monosaccharides, disaccharides, oligosaccharides. Is the formation of rings that result in the creation of an asymmetric carbon, called the anomeric carbon. Anomerism. Table 2.1, Classification of sugars. Pyranose and furanose ring structures. Most monosaccharides spontaneously form ring structures in which the aldehyde or keto group forms a bond with one of its O groups. If the ring contains 5 atoms, it is called a furanose ring. If it contains 6 atoms, it is called a pyranose ring. For glucose in solution, 99% is in the pyranose form. Alpha and beta anomers. Anomerism is the formation of rings that result in the creation of an asymmetric carbon, called the anomeric carbon. Epimers. These are isomers differing as a result of variations in configurations of the O and H on C atoms 2, 3 and 4 of glucose. The most important biological isomers of glucose are mannose, C2, and galactose, C4. Aldose-ketose isomerism. Fructose has the same molecular formula as glucose but differs in its structure. Since it has a keto GRP in position 2 whereas there is an aldehyde group in position 1 of glucose. Glucose, galactose, fructose, and mannose are physiologically the most important hexoses. The occurrence of compounds with the same chemical formula but different structural formula is called isomerism. The important types of isomerism found in glucose are isomerism. D and L is the storage polysaccharide in animals. Glycogen is the chief constituent of plant cell walls. Cellulose is a structural polysaccharide in the exoskeleton of crustaceans and insects and also in mushrooms. Chitin are complex chains containing amino sugars and uronic acids. Glycosaminoglycans. Glycoproteins, mucoproteins, are toteins containing branched or unbranched oligosaccharides chains. Fig 2.1, Structures of cellulose, starch, and mucoproteins. Is the most abundant matter on Earth and also in all living creatures. Water. In living organisms, constitute about 70 to 90% of the body. Water. Acids and bases are defined with respect to their ability to gain or loss Protons. Is a solution that resists change in the pH of a solution when proton or hydroxyl ions are suddenly added. Buffer. A solution that resists change in the pH of a solution when proton or hydroxyl ions are suddenly added is called buffer. The properties of water include the following. Water is the predominant chemical component of all living organisms. Most chemical reactions in the cell take place in aqueous environment. Hydrogen bonds hold the oxygen and hydrogen atoms together in a water molecule. The oxygen of water is very electronegative, while hydrogen is electropositive. As a result, water is dipolar and exhibits slight tendency to dissociate. Biological importance of water. A molecule with electrical charge distributed unequally about its structure is referred to as a dipole. H3O equals H++ O. The strong dipole and high dielectric constant of water enables it to dissolve large quantities of charged compounds. Presence of hydrogen bond also enables water to dissolve many organic molecules that contain functional groups. Water provide environment for macromolecules to achieve stable structure in solution. Is a compound that dissociates in aqueous solution to produce proton, H+, and a conjugate base, A. Acid. The dash is called the acid dissociation constant, Ca, where K is the constant and A is the acid. Equilibrium constant. Dash is a compound that accepts proton in aqueous environment, for example ammonia reacts with a proton to produce an ammonium ion. Base. Dash is a solution that resists change in pH when acid or base is added. Buffer. A dash contains a weak acid and its conjugate base. Buffer. A buffer contains a dash and its conjugate base. Weak acid. A buffer contains a dash acid and its conjugate base. Weak. A buffer contains a weak dash and its conjugate base. Acid. A buffer contains a weak acid and its dash base. Conjugate. A buffer contains a weak acid and its conjugate dash dash. Base. The basic structural units of proteins are called dash dash. Amino acids. Proteins in all species from bacteria to humans are constructed from the same set of dash amino acids. 20. N dash consists of amino group, NH2, a carboxylic group, COOH, a hydrogen atom, H, and a distinctive R group which is specific to each amino acid. Amino acid. The three groups which are bonded to a carbon atom, called the alpha carbon. The isoelectric pH, also called pi, is the pH midway between pK values on either side of the isoionic species. Dash are the basic structural units of proteins amino acid. An amino acid consists of amino group, a carboxyl group, a hydrogen atom and a distinctive R group bonded to a carbon atom, called the alpha carbon. An R group is referred to as a dash dash. Side chain. Amino acids in solution at neutral pH are predominantly dipolar ions, or zwitterions, rather than unionized molecules. There are dash amino acids found in proteins. 20 Table 1.2, the 20 amino acids. The net charge on an amino acid I.E. The sum of all the plus veli and veli charged groups present depend on the pK values of its functional groups and the pH of the surrounding medium. The isoelectric pH, called the pi, is the pH midway between pK values on either side of the isoelectric species. The most important reaction of amino acids is the formation of a dash bond. Peptide. Fig 1.5. Formation of peptide bond. An amino acid unit in a polypeptide is called a dash dash. Residue. The first scientist to determine the amino acid sequence of a protein is called dash dash. Frederick Sanger. Frederick Sanger determined the amino acid sequence of insulin in dash dash. 1953. In the year Frederick Sanger was given a Nobel Prize in Chemistry in recognition of his work. 1958. In 1953, Frederick Sanger determined the amino acid sequence of dash dash. Insulin. Alpha amino acids give an intense blue color while amino acids such as proline give a yellow color. Sequencing of a dash was first carried out by Frederick Sanger in 1953. Protein. The Edman method can be used to determine the first 20 to 30 residues of a dash dash. Peptide. There are dash levels of protein structure. The four levels of protein structure are: primary structure, secondary structure, tertiary structure, quaternary structure. Dash refers to the sequence of amino acids and location of disulfide bridges if there are any. Primary structure. Dash refers to the steric relationship of amino acid residues that are close to one another in the linear sequence. Secondary structure. Dash refers to the entire three-dimensional conformation of a protein and the steric relationship of amino acid residues that are far apart in the linear sequence. Tertiary structure. Quaternary structure refers to the arrangement of polypeptide chains in a multi-chain protein. The roles of proteins in biological processes. Enzymatic catalysis. Transport and storage. Coordinated motion. Mechanical support. Immune protection. Generation and transmission of nerve impulses. Control of growth and differentiation. The only macromolecule without a specific monomeric unit is called dash. Lipid. The word lipid was derived from Greek word lipos. The meaning of lipos in Greek is fats. Examples of lipids include fats, oils, steroids, waxes and related compounds. Biological functions of lipids. Lipids have various important functions in animals. Some of the functions are: lipids are important dietary constituents of food and have high energy value. Dietary lipids are the sources of fat-soluble vitamins and essential fatty acids. They also help in transportation of many water-insoluble molecules. Lipids serve as energy stores. Fat stored in animals as triacylglycerol or triglyceride in the adipose tissue is a potential source of energy. Fats present in the subcutaneous tissues act as an insulating material. V-lipids are important component of plasma membranes. Lipids are involved in various intra- and intercellular signaling processes. Classification of lipids into different groups based on their chemical composition. Simple lipids. Complex lipids. Derived lipids. Fatty acids. Saturated fatty acids. Unsaturated fatty acids. Examples of simple lipids are as follows: fats, oils, waxes. Fats are solid at room temperature because their fatty acids are saturated. Complex lipids contain other amphipathic compounds in addition to fatty acids and alcohol present in simple lipids. Symptoms of essential fatty acids deficiency: growth retardation, poor wound healing, dermatitis, kidney and liver diseases, infertility, depression. Cholesterol is A-alcohol. Monohydric. The 5 classes of steroids are: glucocorticoids, minerals corticoid, carbon structures, progesterone, testosterone. Lipids are not soluble in aqueous solutions such as the blood. True. Lipids are soluble in aqueous solutions such as the blood. False. The following is classification of lipoproteins: high-density lipoprotein, HDL. Low-density lipoproteins, LDL. Very low-density lipoprotein, VLDL. Chylomicrons. The 5 carbon sugars are known as pentoses. Figure 7.1 The structure of ribose sugar. Figure 7.2 The structure of deoxyribose sugar. DNA stands for deoxyribonucleic acid. The acronym RNA means ribonucleic acid. Deoxyribonucleic acid, DNA, is an example of polynucleotide, and it is one of the 2 types of nucleic acids that made up the genetic materials. DNA exists as a stranded molecule in eukaryotes. Double. DNA exists as a stranded DNA molecule in prokaryotes. Single. Figure 8.1, A. Stacking of 2 chains, B. Double-stranded structure of DNA, C. Beads representation of double-stranded DNA. Figure 8.2, The structure of RNA molecule. Is the process by which a replica or identical copy of DNA is made from the parent copy. Replication. Replication is also known as duplication. Is the process by which the genetic messages contained in the DNA molecule are read and copied correctly to produce ribonucleic acid, RNA. Transcription is the process by which the genetic messages carried by RNA are decoded and used to build proteins, enzymes, receptors, hormones, etc. Translation. The process of separating the complementary strands of duplex DNA is called dash dash. Denaturation. When the hydrogen bonds between base pairs of DNA is disrupted by heating, thereby causing the strands to separate, the process is called dash dash. Melting of DNA. DNA solution absorbs maximally at a wavelength of dash millimeter 260. Renaturation is also called dash dash. Hybridization. Dash is the process of reassociation of separated strands of DNA or any two strands of DNA that are complementary in base pairing. Renaturation. The catalysts of biochemical reactions are known as dash dash. Enzymes. The first clear recognition of an enzyme was made by dash and dash dash. Payen and Persas. Amylase was formerly known as dash dash. Diastase. The first enzyme to be obtained in pure form was urease, crystallized from jack beans in dash dash. 1926 James Sumner was awarded the Nobel Prize for urease in dash dash. 1946 A systematic scheme for naming or classifying enzymes was adopted in dash dash. 1972 All known enzymes belong to one of six main classes: oxidoreductases, transferases, hydrolyses, lyases/synthases, isomerases, ligases/synthetases. Dash can be defined as the amount of enzyme required to convert 1 mole of substrate to product per minute under specified assay conditions. Enzyme activity. Dash is defined as the number of enzyme units per milligram of protein, mole/min/mg. Specific activity. Dash is equal to the units of activity per mole of enzyme. Turnover number. Dash is the velocity obtained under conditions of substrate saturation of the enzyme and specified conditions of pH, temperature, and ionic strength. Maximum velocity. The term allosteric comes from the Greek allos, other, and stereos, solid, object. There are major types of regulatory enzymes. 2. The major types of regulatory enzymes allosteric enzymes and covalently modulated enzymes. Metabolites that bind at the allosteric site are called modifiers or modulators. Allosteric effectors. Metabolites that bind at the allosteric site are called allosteric effectors, or modulators. Modifiers. Metabolites that bind at the allosteric site are called allosteric effectors, modifiers, or modulators. Metabolites that enhance the protein's activity are referred to as allosteric activators. Metabolites that decrease the protein's activity are called allosteric inhibitors. A can combine with either the free enzyme or the ES complex, interfering with the action of both. Non-competitive inhibitor. French chemist Henry found that enzyme kinetics was initiated by a bond between enzyme and substrate in the year 1903 is the velocity obtained under conditions of substrate saturation of the enzyme and specified conditions of pH, temperature, and ionic strength. Maximum velocity. Turnover number is equal to the units of activity per mole of enzyme. Allows for direct comparison of catalytic ability between enzymes. Catalytic enzyme. Is defined as the number of enzyme units per milligram of protein, mole/min/mg. Specific activity. Can be defined as the amount of enzyme required to convert 1 mole of substrate to product per minute under specified assay conditions. Enzyme activity. The SI unit of enzyme activity is the Ktal. Is defined as the amount of enzyme required to convert 1 mole of substrate to product per second. Ktal. Table 12.4 Serum enzymes used in clinical diagnosis. The simplest amino acid is leucine. Starch and dextrin are examples of polysaccharide. Active transport is achieved through proteins. Carrier. Sucrose sugar contains and glucose. Fructose. Sucrose sugar contains fructose and dash dash glucose. The phenomenon of osmosis is opposite to that of dash dash diffusion. The degradative processes are categorized under the heading of dash dash catabolism. The most active site of protein synthesis is the dash dash ribosome. Which one is the largest particulate of the cytoplasm? Mitochondria. Which one is the heaviest particulate component of the cell? Nucleus. Glycerol, fatty acid, and dash are the three components of phosphoglycerides. Phosphate. Phosphoglycerides present in the outer monolayer of human red blood cells are dash dash phosphatidylcholine. Sphygomyelins consist of glucose in its structure. False. Membrane proteins that transverse the cell membrane is dash dash integral protein. Ion channels will most likely belong to dash category of membrane proteins. Integral. Major factor that affect the rate of facilitated diffusions are dash dash concentration gradient. Dash is a major component that affect the fluidity of membrane. Cholesterol. Transport across membrane that required energy input is dash dash active transport. Na plus K plus At pos A is a typical example of dash dash active transporter. Damage to Cl ion channel is associated with dash dash cystic fibrosis. Lactose and sucrose has dash in common. Glucose monomer. Sucrose is non-reducing because dash dash it has no free anomeric carbon. Glucose alcohol is dash dash sorbitol. Lactose is a reducing sugar. True. The two components of lactose are dash N. Glucose. The bond between the monomeric units in lactose is dash dash beta 1 to 4 glycosidic bond. The monomeric units in isomaltose is dash dash glucose. The monomeric units are joined together by dash dash alpha 1 to 6 glycosidic bond. Three examples of homopolysaccharides are dash dash starch and dash dash glycogen. The storage polysaccharide in mammals is dash dash glycogen. The nucleotide solely found in RNA is called dash dash uracil. Dash is the nucleotide found solely in DNA. Thymine. Sugar moiety of DNA differs from RNA in dash dash. The sugar is dehydroxylated at position 2 in the ribose of DNA. The nucleic acid that induces hyperglycemia in rat is dash dash alloxan. Nucleic acids of physiological importance not found in DNA or RNA are dash dash uric acid. Examples of essential fatty acids are linoleic acid dash and linolenic acid arachidonic acid. Transportation of lipid from the intestine to the liver is carried out by dash dash chylomicrons. Dash transport cholesterol from the peripheral tissue to the liver. HDL. The transportation of lipid in the blood is carried out by lipoproteins. The precursor of prostaglandins is dash dash arachidonic acid. Which of the proteogenic amino acids is not optically active? Glycine. Glycine, valine, alanine, and leucine are dash dash aliphatic amino acids. Sulfur-containing amino acids are dash dash methionine. Aromatic amino acids are dash dash tryptophan. An amino acid is dash dash proline. Proteogenic amino acids are dash isomer. L. Levorotatory. Is a conjugate acid-base pair solution capable of resisting large change in pH upon addition of small amount of H plus or O dash. A buffer. A polyprotic acid will most likely has dash dash many ionization point. A weak acid will ionize dash in water. Partially. Enzymes are dash in nature. Proteinous. Catalytic activity takes place at dash site of an enzyme. Active. Non-protein part of an enzyme is refers to as dash dash cofactors. Coenzymes, dash and metal group are the three types of cofactors. Prosthetic group. Cofactors that are loosely bound to enzymes are dash dash. Coenzymes. Examples of Mg-containing metalloenzymes are dash dash. Kinases. The classification of enzymes into classes divide them into dash groups. 6. The first class of enzymes belongs to dash group. Oxidoreductases. Enzymes that are involved in structural rearrangement of groups around a compound is dash dash. Isomerase. The value obtained at half maximum velocity is dash dash. Kilometer and holoenzyme will contain dash dash. An apoenzyme and cofactors. Antioxidant enzymes are predominantly found in dash dash. Peroxisome. Which of the following has no effect on the fluidity of the plasma membrane? Asymmetry nature of phospholipids. Which of the following substances are not actively transported through the cell membrane? Sodium ion. Which of the following is not true of phosphoglycerides of cell membrane? They contain glycerol backbone. Which of the following is not true about the property of peripheral protein? Detergents are required to remove them. The factor that regulate facilitated diffusion across membrane is dash dash. The concentration gradient. Cystic fibrosis is a disorder of dash dash. Cl channels. The detoxification of toxic compounds and drug metabolism takes place in the dash dash. Mitochondria. Sodium and potassium channels are example of dash dash. Voltage-gated channels. Cell death resulting from the lack of essential growth factors is known as dash dash. Lesion. Which of these nucleic acid bases can cause glycosuria? Adenine. The condensation product of purine and pyrimidine bases with sugar, ribose or deoxyribose, is DNA. Cyclic AMP is an example of dash dash. Nitrogenous base. The distance between adjacent base pair in a DNA duplex is dash dash. 0.34 nanometers. The distance between neighboring nucleotide in a DNA strand is dash dash. 0.34 nanometers. A substance that reduces the pH of a solution by removing O from solution is most likely a dash dash. Acid. A conjugate acid-base pair will give rise to which of the following? Acidic solution. Protein buffer system is most likely to be found in dash dash. ECF. Increase in blood pH as a result of rigorous exercise is called dash dash. Metabolic acidosis. The pH of 0.0001 mHNO3 is dash dash. 1. When the concentration of acid and conjugate base in a solution is equal, which of the following statement is incorrect? The pH equals pKa. Which of the following is the type of glycosidic bond in cellulose? Beta dash, 1 to 4. The specific optical rotation of equilibrium mixture of alpha and beta D-glucose is dash dash. 18.7 degree. Anomrism is produced with reference to one of the following. Carbon 1 in aldose and ketose. When glucose is reduced, the product is dash dash. Mannitol. Galactose reacting in the presence of nitric acid and heat will give a compound called dash dash. Mucic acid. The bond found in sucrose is between dash dash. Carbon 1 of glucose and carbon 2 of fructose. The following amino acids have right-handed stereoisomerism except dash dash. Valine. Which of the following naturally occurring amino acids is not L-amino acids? Histidine. Which of the following is not a branch chain amino acids? Methionine. Toxic non-protein amino acids include dash dash. Ornithine. Ketogenic amino acids include the following except dash dash. Phenylalanine. Which of the following is not a class of lipid? Simple lipids. The major function of cholesterol is all of the above except dash dash. Synthesis of bile salt. The following are essential fatty acids except dash dash. Linoleic acid. Which of the following is the precursor of prostaglandins? Linoleic acid. Which of the following enzymes are involved in the hydrolysis of triacylglycerol? Cyclooxygenase. The following are true concerning linolenic acid except dash dash. It is synthesized in the kidney. All of the following are correct regarding triacylglycerol except dash dash. Tag are esters of alcohol and fatty acids. The unit of catalytic constant is dash dash. Kettle. The intercept on the x-axis for a Wolf plot is dash dash. km/v max. The slope of Eadie-Hofstee plot is dash dash. km/v max. The slope of Lineweaver-Burke plot is dash dash. km/v max. The intercept on the horizontal axis of Lineweaver-Burke can be used to estimate km/v max. The value derived at half maximum velocity is of an enzyme catalyzed reaction is dash dash. km/v max. The linear transformation of Michaelis-Menten equation will give rise to the following except Lineweaver-Burke plot. Catalysis takes place on dash site of an enzyme. Active site. Regulation of enzyme activity by cleavage of some residue from the enzyme is known as dash dash. Allosteric. Regulation. A non-protein part of an enzyme is known as the dash dash. Cofactor.